Hello, Mr. Allman here, and this is your Allmancast on respiration. Before I get too far into the steps of the actual biochemical process, it's important that we take a step back and think about what we learned earlier in the course about the role of matter and energy in living things. Energy, as you know, has to be continuously brought into ecosystems through sunlight. And photosynthesis is the process that takes the energy in light and converts it into chemical energy. That chemical energy gets stored in carbon compounds that we can call nutrients. Now, all organisms, plants, animals, bacteria, they need to extract energy from those nutrients. And respiration is that enzyme catalyzed process that utilizes that stored chemical energy from those nutrients to be able to produce a molecule called ATP. In eukaryotic organisms like ourselves, the mitochondria are going to be the important cell part in which a lot of this work occurs. But in this video, I want to contrast some of the different forms of respiration that happen both within humans and other organisms. Now, just reflecting one last time on that role of energy, know that not only does respiration produce ATP, but much of the energy from those nutrients actually gets converted into heat. And this is responsible for much of the reason why energy gets lost as energy is transferred from trophic level to trophic level. Now, we've already covered photosynthesis, and there's a curious relationship between the process of cell respiration and photosynthesis. We learned in our botany unit that carbon dioxide and water will be used to synthesize molecules of glucose, simple carbohydrates that will be converted into glucose, as well as oxygen as a byproduct. Now, if you take and flip those reactants and products to the opposite side, you'll see you get the same chemical formula for cell respiration. Glucose, or other similar types of nutrients, are, when in the presence of oxygen, get broken down to make carbon dioxide as one byproduct and H2O, or water, as a second byproduct. Of course, making ATP, utilizing that energy from those nutrients. Now, all of this is very tightly controlled through the use of enzymes. So negative feedback is very important in being able to act upon and controlling those enzymes, making sure that nutrients are only going to be broken down in small little steps, one after the other, to ensure that we don't waste any energy and to ensure that as much energy as possible is going to be released in that process. Now, ultimately, the chemical energy from nutrients will be used to be able to synthesize a molecule called ATP, adenosine triphosphate. Adenosine triphosphate is made up of three main components. There is the adenine, which is a nitrogenous base you should be familiar with from its role in RNA and DNA. Ribose, and together adenine and ribose make adenosine. Ribose, which is a pentose sugar, will then be connected to a set of three phosphate groups. So adenosine triphosphate comes from the adenine, the sugar, and the three phosphate groups attached. Now, that phosphodiester bond, which connects the second phosphate and the third phosphate, is particularly energetic, meaning that when this bond is broken, a lot of energy is going to be released. Now, when that bond is broken in a chemical reaction called hydrolysis, energy will be released, but that ATP, once it's lost, that third phosphate will now become adenosine diphosphate, or ADP. And so in cell respiration, what's happening is the opposite. Adenosine diphosphate and an inorganic phosphate are going to be combined together in a reaction called phosphorylation, using energy from the nutrients being broken down to be able to generate ATP. And so now we have an opposing set of anabolic and catabolic reactions, ATP being broken down to release energy, or ADP being combined with a third phosphate to make ATP, and this bottom part in blue is what is going to be happening as a product of respiration. Now the actual process itself. This red square represents the cell. we have everything in red is going to be in the cytoplasm. What's in yellow is going to be the process happening within the mitochondria. So we're going to start with looking at just a general overview of what's happening. We're going to have a process called glycolysis, which is happening out in the cytoplasm, and all respiration begins there. Then I'm going to compare anaerobic and aerobic respiration. Anaerobic respiration will require fermentation as a follow-up process happening in the cytoplasm, whereas aerobic respiration will continue within the mitochondria. Again, one or the other is going to occur depending upon particularly the presence of oxygen. So we begin with glycolysis. So glycolysis is going to start with a molecule of glucose or a similar nutrient being oxidized. Oxidized means that electrons are going to be removed from glucose. It's also going to get divided in half, making two molecules of a smaller organic compound called pyruvate. Now this process is going to require some energy. An input of 2 ATP is needed in order to be able to actually divide that glucose. However, in the process of rearranging some of those bonds, 4 ATP are going to be produced. Additionally, the electrons that are removed by oxidizing glucose are going to be added onto an electron acceptor called NAD, and it's going to turn NAD into NADH. Here's another image that illustrates the different phases here. The energy investment phase is showing the 2 ATP are being used in order to be able to help break glucose up initially. But in a second energy payoff phase, we're going to take 4 ADP and 4 phosphates, and those are going to be combined together to make 4 molecules of ATP. So we can say that there's a net production of 2 ATP. Now, glycolysis is universal to all forms of life, and it's thought to be one of the most ancient metabolic pathways. And certainly the first cells on Earth would have needed to have some way to extract energy from nutrients. And because this is universal to all forms of bacteria, protists, plants, fungus, animals, this is one piece of evidence that shows that life has a single origin, that life didn't evolve independently many different times, because if life evolved many different times, we might see different types of chemical reactions happening in different things. But because this is a process that happens everywhere in all living things, that shows that this is a type of homologous process, that the ancestor of all living things also went through glycolysis. Again, just to summarize, if we start with a molecule of glucose, 2 molecules of NAD, and 2 molecules of ATP, which is the energy input phase, what we're going to get from that are 2 pyruvate, which are these smaller 3-carbon molecules, 2 NADH, which again received the electrons from glucose, and 4 molecules of ATP. Now from here, depending upon whether or not oxygen is present, then we could either go into anaerobic respiration, which will be followed by fermentation in the cytoplasm, or if oxygen is present, then those products of glycolysis will proceed on into the mitochondria. We'll start with fermentation though. Again, if no oxygen is present. Here, the purpose is simply to be able to oxidize NADH back into NAD+, so that glycolysis can continue. No new ATP is produced during fermentation. Rather, the pyruvate, which is one of the products of glycolysis, is going to be reduced, meaning it's going to accept the electrons from the NADH in order to be able to reset the system to get ready for glycolysis again. There are 2 forms of fermentation that you need to be aware of. One of those is ethanol fermentation, and this is the process that happens in yeast and many types of bacteria. In this process, we start with pyruvate, and again, pyruvate which are produced as a result of glycolysis. Those pyruvate are going to be converted into ethanol. And in the process of converting the pyruvate into ethanol, we're going to release CO2, because pyruvate will first, one of those carbons, so the 3 carbons from pyruvate will be removed to make a molecule of carbon dioxide. And then the 2 carbons and the oxygen that are left over will be rearranged to make ethanol, which is a 2-carbon alcohol. In that process, electrons are going to be removed from the acetaldehyde and are going to be given back to NAD. And so in doing so, that's going to allow for NAD to be able to go back into glycolysis so that more molecules of glucose can be broken down. Now, in humans and animals, particularly in muscle cells, we don't see ethanol fermentation, but instead we see lactic acid fermentation. Here, the purpose is the same. We have NADH, which needs to be converted back into NAD in order for glycolysis to continue and ATP production to be able to continue going on, even if there's a lack of oxygen. So in order to reset that NAD+, the pyruvate is going to accept the electrons from the NADH, and in the process, pyruvate will be converted into lactate. Now, this process is going to be reversible. So in muscle cells, the lactate will build up over a short period of time, but then once the circulatory system returns a supply of oxygen, then that lactate can get converted back into pyruvate, and then the process of aerobic respiration can continue. Now, ethanol fermentation has been utilized for thousands of years by humans as a commercially lucrative biological reaction. We see forms of yeast being used to be able to, for its carbon dioxide, to be able to bake, because those carbon dioxide, when it gets trapped inside the proteins within bread, it's going to cause bread and cakes to rise. When yeast are used to be able to ferment fruit, like with grapes, we can make liquor and wine. The ethanol and the carbon dioxide together are used in the brewing of beer. And we see similar processes going on in things like milk to be able to make yogurt and cheese, or be able to make soy sauce. Just to summarize, again, if we enter into the process of fermentation with pyruvate and NADH, that NADH needs to be oxidized back into NAD+, so that glycolysis can continue. And in the process, the pyruvate is going to be reduced into either ethanol and CO2, like we saw in yeast, or lactic acid in humans. Now, if oxygen is present and we're in a eukaryotic organism like a human, then the pyruvate will not go on to fermentation, but rather the pyruvate is going to enter into the mitochondria. And within the mitochondria, there's going to be a multi-step process. That pyruvate goes through something called the link reaction. In the link reaction, the pyruvate is going to be decarboxylated, meaning that carbon is going to be removed, producing carbon dioxide, which is one of the byproducts of respiration. And then it's going to enter into a cycle called the Krebs cycle, in which, again, more carbon dioxide is going to be produced and released, and some ATP is going to be produced. All the while, we have NADH and FADH2. These are molecules that are holding electrons, and those electrons are going to be transferred to a multi-step process called the electron transport chain. And this is going to use those electrons as a source of energy to drive the production of a very large quantity of ATP. Additionally, we're going to use oxygen, O2, as a final electron acceptor, taking those electrons and getting converted into water, H2O. So some of the important things in here that you need to make sure that you understand are the inputs. So we have an input of glucose and O2, oxygen gas. Those are being delivered by the circulatory system. And then within the cytoplasm and the mitochondria collectively, we are producing ATP, a very large quantity here in aerobic respiration in comparison to the very small amount that we got out of anaerobic respiration. And we're producing also some byproducts, carbon dioxide and water. That carbon dioxide will diffuse back into the blood, where it's going to be excreted, while the water will often remain within the cell to be used for other purposes. Now, I've been focusing here on glucose, but really any type of nutrient can be used, not only sugars. Sugar is going to be the primary form, and there are some organs, like the brain, which rely almost entirely on glucose as a source of energy. But it is possible for different components to enter into this process at different points. Certainly, any type of stored carbohydrate, like glycogen, could be broken down into sugars and used immediately as a source of energy. But fats, triglycerides, can be broken down into fatty acids and glycerol, and those can be used at different points, again, to be able to produce ATP. Proteins can also be broken down into amino acids, and amino acids can be metabolized, but that's only going to be it's only going to happen under a starvation type scenario where all other forms of energy have been used up. And that is the process of respiration in summary. Thank you for listening. Please let me know if you have any questions.